Hello, and welcome back to another student project review by Group Devi1. In this video, we would like to discuss about our student project regarding the topic of extended spectrum beta-lactamase. To give better learning experience, we decided to divide this presentation into a couple of outlines, which the first one will be the introduction, continued by the main topic, and finally the summary. Right next after this, Bintang will give you some insight on why we choose this topic and other general information regarding our student project.
Hello, my name is Putu Bintang with last name number R033. First, I will explain what the background information. So, beta-lactam are a type of class from antibiotics. The first beta-lactams are penicillin, which was found by Alexander Fleming in 1920s. Bacteria give response to beta-lactam by producing beta-lactamase, an enzyme to inactivated beta-lactam antibiotic. Along with time, bacterial resistance evolved become extended spectrum beta-lactamase or ESBL, which is the new resistance for bacteria to resist most of antibiotics. Some of the bacteria are Escherichia coli and Klebsiella pneumonia. Next, the global prevalence of ESBL producing Escherichia coli in the world reached 16.5%, while in China it's reached 62.8%. Meanwhile, Klebsiella pneumonia have higher emergence rate in Indonesia compared to other developed countries. In our student project, we will discuss the ESBL and its effect on the development of current antibiotic development. Next, the problem formulation. 1. What is beta-lactam antibiotic? 2. What is beta-lactamase? 3. Who produce beta-lactamase? What is beta-lactam resistant bacteria? 4. What is extended spectrum beta-lactamase? 5. How is the effect of extended spectrum beta-lactamase on the current antibiotics development? Next, the objective. The general objective are to learn and to understand better about extended spectrum beta-lactamase and its effect on the current development of antibiotic. The specific objectives are in accordance to the problem formulation. Next, the benefits. The benefit is to show and to learn the clinical science regarding the extended spectrum beta-lactamase.
My name is Carin and my last digit number is 016. Penicillin was reported as the first beta-lactam antibiotic and became the basis of modern antibiotics. It all started when Fleming was growing a culture of Staphylococcus and observed that penicillin notatum contaminated the dish. Unfortunately, Fleming did not expand his work into clinical studies because he was unable to purify penicillin and characterize its activity. Then, in 1939, scientists at Oxford started a new antimicrobial study using Fleming's articles as a basis. However, Florey and Ernest Chain continued the study and did trials first in mice. This observation provided evidence that penicillin has potential as a life-saving drug. By early February in 1941, the Oxford team was able to verify enough material to carry out experiments on humans, the results showing an imposing effect without any toxic side effects. Then, the Oxford team recognized the potential of penicillin to treat soldiers and civilians wounded during the war. Since the discovery of penicillin, thousands of penicillin derivatives have been discovered, including cephalosporins, carbapenems, monobactams, and others. That's all from me. Thank you.
Thank you for the opportunity. My name is Adjani Sayidina Pakpahan with last 3 digits of student number 031. Beta-lactam antibiotics are one of the most widely used antibacterial medication classes globally. From a biochemical standpoint, these medicines share a 3-carbon and 1 nitrogen ring, which is highly reactive. This class consists of penicillins, cephalosporins, carbapenems, monobactams, and inhibitors of beta-lactamase. Mechanism of action. Bactericidal activity is the mechanism of action of beta-lactam antibiotics. By isolating the trimpeptidase involved in cross-linking peptides to generate peptidoglycan, beta-lactam antibiotics impede the final step in peptidoglycan formation. Mechanism of resistance. Some of these are inactivations via beta-lactamase synthesis. There are many uses of beta-lactam antibiotics, for example, penicillins, which is used for Streptococcus pneumonia and meningitis, carbapenems, which can be used for urinary tract infections, cephalosporins, which consists of 5 classes, for example, the first generations, can be used for serious infections of skin and soft tissues, and lastly, monobactams, which used for pneumonia.
Okay, thank you for the time. My name is Nyoman Gede Sujana with the last 3 digits of number 011. So today I'm going to tell you about the history of beta-lactamase. Okay, next slide please. Beta-lactamases are ancient enzymes that exist even in lack of the pressure of therapeutic antibiotics. In 1940, when Abraham and Change reported that the bacterial enzyme was able to destroy penicillin. Recent explorations have identified functional beta-lactamase-like activity in some locations that found beta-lactamase sequences that are unknown. They also found ubiquitous DEM beta-lactamases from 30,000 years old permafrost sediments east of Dawson City, Yukon, Canada, in a slightly younger sample of cold seep sediments of Addison Seamount, Papua New Guinea, encoded DEM family beta-lactamases. On the next 2 slides, you guys can see the table of the dates, organisms, and locations of the first of series of beta-lactamase producing isolates with long-term clinical significance.
Thank you, Satria. Welcome back to me. My name is Gede Saputra with last 3-digit number name 018. As we discussed before, beta-lactamase is an enzyme that is responsible for the beta-lactam antibiotics resistance. They achieve this feature by changing the structure of beta-lactam in the form of promoting hydrolysis of the beta-lactam rim present in almost all class of penicillin. Richard Ambler classified this beta-lactamase into 4 classes, which is class A, B, C, and D, based on their sequence, motif, and later, based on their structure and mechanism of action. They can also be grouped into 2 main groups, which is the serine beta-lactamase or SBLs and beta-lactamase known as MBLs. The SBLs consist of class A, C, and D beta-lactamase because they have similar amino acid motifs, shown orange in the picture, a unique serine lysine motif that is important for the work mechanism. After activation by basic informant dehydrogenation, the serine will initiate a nucleophilic attack on the carbon-carbon C7 of the beta-lactam ring, creating an acyl-enzyme covalent bond that is fragile to hydrolysis by a water molecule called the acylating water. And then there's MBLs, which only has class B beta-lactamase in it, but what differs them from other beta-lactamase is the presence of zinc ions, shown gray in the picture. Just like the SBLs, MBLs begin their action mechanism by activating the zinc ion with the help of substrate binding, in this case beta-lactam antibiotics, then it attack upon the fragile cis carbonyl, causing 2 possible solutions. First via a protonation at C2 in the beta-lactam ring by hydrolysis, and second via protonation at N4 atom of the beta-lactam ring by hydrolysis.
My name is Putu Krishna Arsawardani Putri with last 3-digit number 027, and I will explain about evolution in some beta-lactam resistant bacteria. The most common ways bacteria acquire resistance for beta-lactams are through production of beta-lactamase enzyme and mutation in the penicillin binding protein. A few examples of beta-lactam resistant bacteria are shown on the slide. First, Streptococcus pneumonia. Streptococcus pneumonia is an extremely recombinant pathogen with a high genomic plasticity. Pressure from host immune system and antibiotics can lead to emergence of new genotypes that have ensured its persistence over time. Next is Escherichia coli. Beta-lactamase producing E. coli include variants of TEM1, TEM2, SHV1, or C2XM enzymes. The newly emerging and rapidly disseminating ST131 E. coli caused inevitable clinical impacts on resistance to beta-lactam antibiotics. It is highly virulent due to its numerous genes that encodes various virulence factors. Next is methicillin-resistant Staphylococcus aureus, or MRSA. MRSA strains can evolve from being low-level to high-level beta-lactam-resistant strains. Mutation in RNA polymerase subunit beta, or subunit beta prime, can lead to production of high-level beta-lactam-resistant strains.
Hi, my name is Khadra Kamila with the last 3 digits number 009. I would like to explain about the impact of beta-lactam resistant bacteria in body and in bacteria. So, beta-lactam is one of the antibiotics most often used as an effective treatment against infectious disease. However, it is well known that bacterial mechanisms against these therapeutic agents are also evolving and slowly increasing at a worrying rate. The increasing resistance of bacteria has a considerable influence on the process of handling patients in hospital and increased morbidity and mortality, which will make patients have to receive treatment for a longer time and also cause complication in the treatment process. Antibiotics belonging to the beta-lactam group tend to be selective inhibitors of the cell wall bacteria and thus are active on bacteria that are in the growth phase. The bacterial resistance occurs due to the presence of lactamase mutases, especially amino acids that are in active site. It causes ineffectiveness in binding, resulting in reduced inhibition. Prevention of beta-lactam ring degradation can be done in several ways, namely in combination beta-lactamase inhibitor compounds, for example, sulbactam and tazobactam.
Thank you for the opportunity. My name is Geg Mas Anantalakshmi with last 3 digits of name number 001. Today, I will give a general overview about ESBL and its diversity. Ambler categorization classifies ESBL as serine lactamase of class A. ESBL is used to describe enzyme produced from TEM and SHV. ESBL are identified by first, ability to hydrolyze expanded spectrum cephalosporin, second, their sensitivity to clavonolate, third, their sensitivity to carbapenem. This characteristic led to the usage of clavonolate as a detector and carbapenem as therapy agent. Diversity of ESBL consists of first, 10 beta-lactamases, which is a derivative of 10-1, identified in Athens, Greece. It is named after the patient Temonera, who provided the first E. coli sample. Second, SHV beta-lactamases. It is a sulfide variable that frequently shown in Klebsiella pneumoniae. Third, C2XM beta-lactamases, which is active on cefotaxime and was first obtained in Munich, Germany. Over the last 15 years, it has spread rapidly and globally, and it outperformed TEM type and HHV type ESBL.
Thank you for the opportunity. My name is Putu Isyundra last digit number 003, and I'll explain about the transmissions, infections, and also risk factor of ESBL producing bacteria. ESBL infections can spread by simply touching someone or leaving the bacteria on the surface where others touch where the bacteria live. ESBL is described as a hospital-acquired infection. It is carried over from reservoirs such as food, water, and medical facilities. Mostly, they are spread by doctors, nurses, or other healthcare professionals who touch people, objects, or surfaces in facilities where the bacteria live. Next, ESBL producing bacteria colonizations in the vast majority of instances is asymptomatic and does not result in infections. But if the bacteria succeed in infecting a person, the implications can be severe. Conditions that can be caused by ESBL producing bacteria include urinary tract infections, pneumonia, skin infections, and also diarrhea. The last one is risk factor. People with urinary catheterizations, central venous catheters, nasogastric tubes, drainage, and also artificial organs were risk factors for ESBL productions. And also, the use of antibiotics was generally a significant risk factor too.
Thank you for the time. My name is Devi Widhi Prastiti with last registered number 028 and I will explain about ESBL testing, prevention, and treatment. Testing, in order to know the existence of ESBL, requires a screening and confirmation test. Screening methods could be done by disc diffusion. Using more substrates such as ceftazidime and cefuroxime can increase detection sensitivity. Besides that, susceptibility tests with antimicrobial deletion with the result of antibiotic concentration is at screening level also show the production of ESBL. To confirm it, broad microdeletion can be used with the sign of twofold serial deletion declining in the condition of minimum inhibitory concentration. Also, combining the disc with clavulanate can also confirm the production of ESBL. To prevent the spread of ESBL producing microorganism, control management by hospital personnel should be priority by having hygienic medical kits, avoid irrelevant use of invasive medical tools, and washing hands regularly. Regardless of being common pathogens, there are still a few available treatments for ESBL. First is carbapenems that have a role in decreasing selective pressure amidst gram-negative bacteria. Next are fluoroquinols and cefamycins that is as effective as carbapenems. And last is beta-lactam-beta-lactamase inhibitors combinations that might be able to restrain ESBL producers due to their morphology as negative gram bacteria.
My name is Danissa Dita with last digit number 005 and today I will explain about developing antibiotics for ESBL. First, fosfomycin. Some studies showed that fosfomycin has maintained activity against the ESBL. Second are the BL-BLI combination, therapy of beta-lactam inhibitors combination as an alternative treatment. There are two examples of BL-BLI combination, ceftolozane and tazobactam, that already been examined the efficacy and also obtaining a good result. Second, ceftazidime and avibactam. Studies also concluded that this combination can be used as alternative treatment. Next one, the new BL-BL combination. There are sulopenem that according to the clinical trial has ability against ESBL. Second, taniborbactam and cefepime with in vitro activity showed potent activity against Enterobacterales. And the last one are combination between N-metotazobactam and cefepime that the in vitro study showed that this combination is as effective as carbapenem. Last one are clazomycin that show equivalent activity against ESBL producing and non-ESBL producing E. coli, and there are still many other new antibiotics being tested.
Hello, let me introduce myself. My name is Librawan with 3 last digit of name is 029. I want to present about ESBL in the future. ESBL is increasingly worrying in the medical world in the future. The emergence of epidemics, multi-resistant in clinical implication, and difficulties in laboratory diagnosis are important aspects that must be faced. In an epidemic, the high level of ESBL producer in the developing countries is a worrying situation in the future and needs to be addressed to stop the epidemic. The emergence of multiple resistances in clinical implication is also quite worrying in the future. However, scientists have innovated on a shorter and easier SBL detection tool, namely matrix-assisted light desorption ionization time of flight.
Thank you for the opportunity. My name is Marvella and my last 3-digit number is 007. Today I'm going to continue my friend's explanation about the conclusion and suggestion. So the conclusions are: first, beta-lactam antibiotic is one of the most widely used antibacterial medication classes globally for certain bacteria. Second, beta-lactamase is an enzyme that degrades the beta-lactam as of antibiotics. Third, beta-lactamase is produced by bacterial cells with beta-lactam resistance. Then, extended spectrum beta-lactamase or ESBL is a serine beta-lactamase classified as class A by Ambler molecular and structural categorization. Then, ESBL has become one of the antimicrobial resistance infections that are concerning by increasing mortality and limitations in diagnosis. Lastly, it is highly suggested for medical students to find out about what extended spectrum beta-lactamase is and its effects on the current development of antibiotics to make an advance to the next stage by solving the ongoing problems regarding the ESBL. That's all from me, and thank you.